Howdy, my name is Eros Aranda. My lab partner is Roberto Blanco, and this is our lab video report on gram, acid fast, and capsule staining, capsule and endopore staining. So introduction, microscope, obviously is very important. We use it all the time. Microbiology, it's in the name. We can't see these super small microscopic organisms with our eye. So we have to actually learn to be able to use microscopes to actually see them. Thing is, these bacteria, a lot of the times we can't see them without having to do staining techniques. So here we can do staining, simple staining, um, other methods of staining that will help. So materials and methods. Um, Obviously media, we have to use different types of plates in order to actually get our cultures and get our be able to place them onto the slides. It's just kind of the cultures from which we actually got our cultures. And our cultures themselves, we used Staphylococcus aureus, Bacillus cereus, Klebsiella pneumoniae, Mycobacterium sp, and Bacillus subtilis. Now for the procedure. So procedure, for the sake of time, we didn't go too crazy deep into this, but it's just important to understand that each staining procedure does go very differently. For example, gram staining uses crystal violet, iodine, alcohol, and safranin to be able to distinguish between gram positive and gram negative. Um, Acid fast staining, we use carbofusion, acid alcohol, and methylene blue. Capsule staining uses crystal violet and copper sulfate. Biggest thing from this one is no heat fixing. No heat fixing. If you heat fix a capsule stain, there's a possibility of causing the capsule to actually burst, and you no longer have a inside capsule and you can't see it. And then for endospore stain, we use malachite green with heat and a saffron canner stain. All of these we ended up observing under oil immersion. So using 100 objective and 10 times magnification, it went to 1,000 times. So this is something beyond the human eye limit, and so lucky to be able to actually use it in the lab. Now for gram staining, just sort of going to fly through this. On S. aureus, it's looked like gram positive cocci. The only thing, it was kind of hard because we kind of layered it together real thick. We couldn't see it too much. For Bacillus cereus, this one's actually a lot easier to see because it was gram-positive Bacillus. There was a lot of those little small spheres floating around, so it wasn't too easy to tell what it was. But in the end, we decided, it was going to be gram-positive Bacillus. And a big thing also, there's that large red clump there on the right side. Most likely, it's just dye that wasn't rinsed off. But overall, we were able to distinguish what it is, but it was pretty difficult to see just due to the thick layering of it. For S. cypherium, you can actually see there's a lot of gram-negative bacillus because you see that long rod-shaped pink bacteria. This one we actually did a pretty good job on because you can actually see good separation between the bacteria. You're able to distinguish individual bacteria. Now for the gram staining of our unknowns. So on unknown 271, it was honestly, I'll be honest with you, really difficult to see. Biggest reason because this one, I even noticed in our culture, it's really small colonies. It's real hard to actually grab some. So it wasn't too easy to see, but it was known to be gram-negative bacillus. Now, looking at unknown 131, this one is a lot better to see, and you can see it was obviously gram-positive cocci. This one was layered on good, correctly, and there was some clumping in some areas, but it was still pretty easy to see the bacteria. I know you can see that large pink dot on the side. Once again, this is probably just dye that wasn't rinsed off correctly. But overall, it didn't really affect the actual quality of the stain. And for acid fast staining, so interesting enough, the instructions say we use K pneumoniae for this one, but in reality, the prepared slide had M. gordonii and Corynebacterium specialis. The biggest thing for acid fast staining is it just tells you if you have bacteria with waxy cell walls. So there's only going to be there's only going to be two outcomes. Either you have bright purple, which means you have a positive, so you do have waxy cell walls, or if it's a blue, which means it's negative, so you don't have waxy cell walls. You can see in our positive one, here, this image in the middle, we can have really bright purple, long rod-shaped structures. That indicates to me it is positive. That's the M. gordonii. Now on the right side, you see, oh, these are just blue. There's no purple in that at all. So that means the stain didn't stick with it. So that's the negative Corynebacterium SPP. Now for capsule staining. This one is a really interesting procedure as they both followed the same procedure, yet they were done differently to begin with. So in the crystal violet, rather than using water to grab the actual K pneumoniae, we use crystal violet. So this means the bacteria grabbed on crystal violet and then was smeared. This made it really, really difficult to actually see the individual cells. This image there left labeled crystal violet. The individual cells are really like you can tell where they are, but it's a really just thick image, layered cells everywhere. The good thing is you can see in the bottom right, there's like a pretty large cluster of little white spheres, which means there is unstained bacteria there. Now, once again, capsule staining, we use it to actually identify bacteria that have capsules and determine the size and shape of the capsules. For the deionized water, this one, there's fewer cocci all around, but it was a lot easier to see because we use copper sulfate, which is a decolorizer. So the issue with the crystal violet is one, the crystal violet, there was a lot of crystal violet which the bacteria set into. So it was rather hard to actually see it. Like the crystal violet like seeped into the cells and wasn't even rinsed off with the decolorizer, the copper sulfate. In the DI though, that one we actually did it properly. We smeared it with the DI, so we was actually able to see a thin layer. We stained it with the crystal violet, and then we rinsed it with the decolorizer. This made it so easy to actually see the individual cells to the point where you could even count the entire slide. In endospore staining, this one was honestly a really interesting one because although we did mess up a bit, we still ended up succeeding in this one. So bacteria with endospores you'll either see two things. You'll see endospores that are a little greenish and other cells will appear red. So in this one, you can actually see right here in the middle, you can actually see a really tiny endospore that was stained. Although in this case, it doesn't look too greenish in the photo, it looks rather bluish. This one, you can see that there's actually an endospore present in B subtilis. This one was really, really interesting because in this entire slide, we only used one drop of the sample to spread around. This was a mistake on our end because we were supposed to actually use four. Because the thing with this is the colony was actually so dilute, it's really hard to actually find one when you stain. And when we actually did the actual staining, put the endospores and all that at the end, we actually ended up seeing that tiny one. Yeah, this one thing that I really liked is we used malachite green in order to actually see them. And side note, not really related to this, malachite green is also used for fishes. It's a good bacterial killer. So I don't know, that's a fun fact, I guess. But yeah, no, this one was a really interesting one that we weren't able to see too much, but we ended up finding one endospore in that entire site. Actually, two. There was one somewhat far off. Over in conclusion, in this exercise, we were actually able to perform gram acid fast capsule and endospore staining to differentiate and identify various bacterial species based on structural and chemical characteristics. Without getting too far into reading all that, honestly, this was a great lab in which we were actually able to test each of those staining techniques. They all differ widely in what their methods are, what they want to looking for, what they're looking for, and how they're done. For example, I know in the endospore, this one we actually had to have the sample suspended over boiling water to get that heat. While compared to gram staining, it's just simply putting dyes, rinsing, putting another dye, rinsing, putting another dye, rinsing. These are all completely different other methods, and we actually practiced it this time for our first time. So each of these produced clear and accurate results, in which each of them we got what we were looking for. This lab overall enhanced our microscopy skills, deepened our knowledge in bacterial morphology, and demonstrated the importance of staining and microbial identification and classification. Overall, I'd say this lab was a success. We learned everything we needed to, and with time, we would definitely get better, fix all the small mistakes we had. And yes, um, thank you very much for listening.